I am a research assistant in the lab of Dr. Richard Cody, who is formerly professor of pathology and urology at USC and currently the chair of chair of pathology at University of Miami. And he's had a long-standing interest in developing technologies to detect and characterize micrometastasis. And in the latest of that development, here, let's do that. In the latest of that development, in collaboration with California Institute of Technology, with their Department of Electrical Engineering, we've developed a membrane microfilter device to capture and characterize circulating tumor cells from blood. So this has arisen from, as Dr. Jans indicated, this has arisen out of the fact that circulating tumor cells have emerged as a potential surrogate biopsy that would represent metastatic disease in cancer. So circulating tumor cells, we're discussing cells that are shed from the primary tumor. They enter the systemic circulation and they attach at other sites and give rise to secondary, tertiary tumors, the basis for metastasis. And as of late, there's been a number of studies that have been done that show that changes in CTC count throughout the course of therapy are predictive of response to that given therapy and the patient's overall survival. Now, the capture and characterization of these cells is no easy task. These cells are extremely rare. These events occur as low as 1 or 2 cells in 1 milliliter of blood, which contains billions of normal blood cells. So being able to sensitively and specifically identify these cells is very difficult. As of late, there's been some technologies that do so. The majority of them do so affinity-based, I can never say that word, platform, which is they use some sort of support system which is functionalized with antibodies that are targeted against a certain marker that is expressed exclusively on tumor cells. And the problem with this is that the expression of those antibodies, of those markers that are used, is variable. So, for example, in prostate cancer, which most of these systems use EpCAM to enrich these circulating tumor cells. In prostate cancer, EpCAM is only expressed on about 70% of prostate cancer tumors. So you have a whole subset of patients for which technologies that are currently available are limited. So what we've come up with as an alternative to this is a size-based method of isolating circulating tumor cells, which is we use the well-known concept that circulating tumor cells are characteristically larger than their normal blood cell counterparts. And so we've developed a microfilter with precisely controlled pore sizes to be able to allow the smaller normal blood cells to pass while we capture the circulating tumor cells, which you see one in our model system is captured by the device right there. Here's an image of the complete fully assembled device. The microfilter would be contained in between 2 slabs of PDMS material, which is housed by an acrylic housing device, and it has a lure lock attachment for blood samples to be squeezed through using a syringe. And in this next one, I have an actual video which would demonstrate this. So what's happened is the blood has been diluted 1-to-1 in PBS, and it's been fixed in a final concentration of formalin for 10 minutes, and then immediately after that it's subjected to the filtration process using steady low pressure. And you can take note of how easily the blood passes through the device, and how quickly the blood also passes through. This is sort of the last of that same sample. We can process an entire sample, which would be anywhere from about 7.5 mils of blood to 10 mils of blood in about 90 seconds, which would be a fundamental difference from the currently available technologies, which take hours to do so. So, once we have those cells isolated, our means of identifying the cells, we use immuno fluorescence. As you can see here, this is just a montage of photos in our model system. Cytokeratin is the classic marker that we use. It's widely expressed on epithelial cells. So you see that in green with the blue DAPI. And we've also developed the ability to do multi-marker characterization. In this panel, you see the cytokeratin in green, PSA, prostate-specific antigen, in red. These cells are isolated from a metastatic prostate cancer patient from a University of Chicago, and then here would be the merge of the three. So our ability to do that multi-marker characterization is really going to be the basis for how we see this technology to be potentially applied in the clinic, which would be to get a blood draw after diagnosis and do CTC characterization using not only what's available now, which is to be able to quantify, but to actually look at different targets. So, for instance, a drug invented or a drug that's purported to have some sort of effect on protein X. We can actually look at that protein in our multi-marker panel, and we can assess, based on the expression characteristics, what type of treatment would be most viable for that patient, and then continuously monitor the characteristics of the CTC and how they change throughout the course of the patient's therapy, and be able to modify the patient's therapy in real time, which would constitute a major move towards personalized patient management, which is not, using a larger rubric to treat patients, but to actually use the patient's biomaterial themselves to be able to assess which course of treatment would be best.